On the agenda, if you notice that now we're going to move into a talk about some treatment options that are available to people that would not necessarily be, I would say, acceptable on a mainstream point of view, but they are evidence-based, meaning that there's a scientific reason for why we are giving this kind of information out to you. My personal reason is because as you know, my daughter was very, very ill, and antibiotics were not working to get her well. You know, she would have ups and downs, but overall, you know, a year into the treatment, we were hoping that she'd be back to college and back to doing things. And so by a very chance experience, I met an individual who was using something to treat his own Lyme disease. And as a result of that, this was a Rife machine. And I said, what is a Rife machine? And he tried to explain it to me, but then I needed to do what I do, which is, of course, do a deep dive into the physics of Rife machines, which is a very scary thing. Okay, so, but the research that I did led me to realize that this is something that there is real scientific basis for. And then, of course, because I am a microbiologist and a scientist, I said, before I'm going to use this on my daughter, I'm taking it into the lab, and I want to see if it actually does what, you know, what it would appear to do. So what I'm going to present is just the results of my research on that, and I'm going to tell you that my daughter uses a Rife machine, if you want to call it that, to help get herself well. So first, just a little bit about what it is. I want to give you a little bit of background on the science, but not too much, because, like I said, it's physics, and I know that is a bad word, just like math, for some people. So just to kind of talk about what it is, we're talking about waves here, electromagnetic radiation, of which there are many different kinds. But a wave is simply what my physics colleague used to say is a disturbance that propagates through a media. And you can see a wave. You can see actually several waves there. So the disturbance is this continual up and down. And when you stop to think about what this is, the light that is shining on you right now is coming to you in waves. My voice is coming to you in waves. When you heat up water in a microwave, that is because of waves. So that's what those are in a practical sense. It's a form of energy, and really not a whole lot else. So here are two waves, one of a longer, these are characteristics of wave. How to characterize them is by their wavelength, which is defined as the distance between, you know, the tops of the waves or the bottoms of the waves. And you can see the red wave has a much longer wavelength than the shorter wave, and we can use that to define something called frequency, which is simply a measure of how often, you know, that wave cycle goes by every second. So that is something called a hertz, named after probably somebody named Hertz. I don't actually know that, but that is often how we refer to these. So to take that further, what do these waves actually do for us? So realize that every object, every cell in your body, everything in this room, animate, inanimate, has a natural frequency that things are vibrating at. And when I mean vibration, you know, I'm sure everybody's starting to sort of feel that up and down motion going, and you want to get into it a little bit. That is something called a vibration. So when that object is disturbed by a force, that natural vibration will start to increase if you know what the natural vibration is. So you can amplify that. And what will happen is those oscillations will just, as it gets amplified, continue to grow until it becomes critical and the energy has to be released in some way. And generally speaking, it's released in either it's let off by light waves or sound waves, or the object will break. So an example of that, which is you know, an example used in every physics class ever of vibration, what oscillations can do, is the Tacoma Narrows Bridge, which in 1940 was a bridge that spanned across the Tacoma Narrows. It was a suspension bridge, and I've never been to Washington, but I believe that this is an area where winds go howling by, and that suspension bridge was known to oscillate, especially on windy days. And then on one particular windy day in 1940, because the oscillations got worse and worse and worse and continued to grow, the bridge actually broke. It broke right, it fell into pieces into the river. And another equivalent is Ella Fitzgerald, the singer, is famous because she was actually able to use her voice, the sound waves, to shatter glass. So it is sort of that same principle. That's how energy gets let off by things breaking. Okay, just the fact that you can hear my voice is energy being let off as sound. So each pitch that I give you is a different frequency of noise. So what we know in biology is that many things inside of cells have natural frequencies. And we also know that cells communicate not only by sending chemical messages to each other, but by sending messages through vibration. So in other words, they're sort of using a cell phone, if you will. So they can actually do that. They can vibrate. That energy goes over to a different cell, picks up that energy, and the word spreads, so to speak. So they're all kind of yelling at each other. We know that they do this in biofilms. They do it through chemical messaging, and they also, bacteria, which Dr. Ehrlich can come back up and talk about that, but they do something called twitching motility, which just means they twitch, and that is a vibrational signal as well. And so when it comes to using this as a way to, as a treatment for something, it's used often as a way to, it's used in a couple ways, right? So the idea is you use frequencies to enhance some natural process or to stop some natural process. And on the stopping side, what is it that those vibrations could actually be doing? And the answer is cells, especially their DNA, which is their genome, their genetic material, vibrates at a natural frequency. And because everything's DNA is different in terms of the structure of it, they are all vibrating at different frequencies. And if you can match that frequency and induce that sort of lethal type of vibration, either the cell will break apart, the cells also listen to what the frequency that their DNA is vibrating at, and at certain frequencies, the cell realizes the DNA is not vibrating at its right frequency, it's damaged, and that is a signal to the cell to actually self-destruct as well. So it's, the idea is, and the other thing is because they're sort of communicating through this vibrational cell phone approach, you could actually break the communication cycle by inducing out external fields in the process. So this has been shown scientifically. This work was actually done by my colleague and collaborator at Skidmore College in upstate New York, Dr. Anthony Holland. Dr. Holland is interested in cancer, realized that there are FDA-approved cancer therapies that are based on this type of thing. This is a series of photographs that he actually filmed working with cancer researchers at Thomas Jefferson University, where what you see is 3 leukemia cells under the microscope, and through lots of experimentation, he was able to find specific frequencies that cause those cells over time, as you can see, right, 3 intact cells, then one by one, they break apart, and by the time the series is over with, all 3 of those cells had actually been destroyed. They were broken apart. That is by using what he called at the time PEFT, which stands for, what does that stand for? Plasma emission field treatment, okay, which is because he was using an emitted field, which I'll show you how that works in a second. This was under the influence of those waves alone that he was able to exhibit this effect. And to take that one step further, he was also able to show that in the presence of chemotherapeutic agents, right, chemotherapy drugs, that this effect was even more enhanced. It was increased, the effectiveness of the drug was increased under the influence of these electric fields. So then that brought me to me. So I'm being advised that here's this weird machine called Rife that is going to help my daughter get better. And I, being the skeptic that I am, said, yeah, right. And then did a little bit of research and said, well, all right, maybe there is something there. And then I said, I got to see it. I got to do it myself. And so working with Anthony Holland, who had the actual machine, the technology, which is simply a wave generating device, a frequency generating device, hooked up to a glass tube, which is filled with helium. And the helium is in the form of a plasma, which is the 4th state of matter, for anybody who cares about that. And so when you beam the frequencies into the, that helium, what happens is an emitted field of harmonics of that frequency. So what goes in is one frequency, what comes out are harmonics of that frequency. And so what you can see is my experimental setup in my lab, so coming from the tube, the helium plasma, with the sound waves going in, coming out as a field. You can actually see that we could measure those, the field coming out, and you can see cultures of bacteria that were being maintained at a constant temperature that were being exposed to those fields. And so these are just two graphs of the research. The first bacteria we decided to work with was a bacteria named MRSA, which stands for methicillin-resistant Staphylococcus aureus. And this is a bacteria that is an emerging problem in healthcare settings because for a lot of reasons, one of which is it's resistant to antibiotics. And so what you can see in the top panel is the exposure of the bacteria to the antibiotic without any external applied fields. And the blue line is what happens when you put bacteria in culture media. They grow, they increase in number very rapidly, so you can see the rapid rate of growth. And then when you add the antibiotic, in this case the antibiotic was methicillin, you can see a really significant reduction in the growth rate. And you might say, why is methicillin-resistant bacteria showing, it looks like it's stopping their growth? And the answer is, not all methicillin-resistant Staphylococcus aureus is actually resistant to methicillin. In some cases, it can actually control their growth, which I didn't know until I started doing this. So the point is, when you then exposed the bacteria to what OPEF stands for, oscillating pulse electric fields, but think of it as sound waves, the harmonics of sound waves. One thing to notice is that the overall growth rate of the culture was significantly reduced. And you can tell that from the scale on the left-hand side. And I apologize for this, but my undergraduate students who presented this research made these graphs. So that's why it would be better if they were on the same the same scale, but realize that the lower graph, although the two lines look the same, what you're seeing is overall the rate of the bacterial growth was reduced, and interestingly enough, in the combination state of sound waves and the drug methicillin, the growth rate is going down. So if the growth rate stays steady, that means the treatment or whatever is bacteriostatic, meaning that it's inhibiting the growth of the bacteria. If it goes down, it means it's killing it. The number of cells is being reduced. So that was the first effect we saw. And then we switched to, well actually, so I just want to point out that this effect is not new. The article on the left is actually authored by William Costerton, the father of biofilms, who noted something called the bioelectric effect, that it was able to inhibit bacteria in biofilms. The one on the right was published 3 years ago in 2014, which essentially shows that what they're calling pulsed electric fields was able to eradicate a bacteria, Acinetobacter baumannii, which is found, is a biofilm bacteria found often in burn wounds, very complicated, very difficult to eradicate. The sound waves alone were able to do those. So then we decided to try this again with a bacteria that produces a biofilm. It's Pseudomonas aeruginosa is the name of this bacteria. It is the first biofilm producing bacteria. Is that right, Dr. Ehrlich? Is that correct?
First pathogen that was characterized.
Okay, the first pathogen that was characterized as producing a biofilm. It is well known for it's renowned for its resistance to antibiotics. One of the tools it uses is an efflux pump, which is when antibiotics go into the cell just spits it all back out again. Okay, but that pump is run on electricity. All right, so the efflux pump that spits out the antibiotics is run on a electrical signal, if you want to think of it that way. And so looking at the graphs, forget the graphs, look at the message at the bottom. So when you were talking about the antibiotic alone, without any kind of exposure, wave exposure, so notice the green line on the top is the control, meaning this is what happens when you put bacteria in media. They double in number for a period of time. And then when you added the antibiotic, in this case the antibiotic was ampicillin, notice that the growth rate was pretty much the same. It wasn't killing, it wasn't growing, it was just staying the same. So ampicillin was able to reduce the growth rate of the bacteria, right, to a steady state, whereas with when you added when you did this on the other side without the antibiotic drug, that it turns out that the oscillating pulsed fields were inhibitory to the same extent as the antibiotic. So that said that this treatment, this idea of sitting in front of a glowing tube, was able to reduce the rate of growth of Pseudomonas aeruginosa to the same extent as the antibiotic ampicillin. And then when you looked at it this way, when you combined them, what happens in both of these both of these graphs is that when you combined the two, when you combined the antibiotic and exposed it to the electrical fields, notice what happens to the growth rate. So it starts out, notice at 25, but the growth rate is going down, meaning the number of cells is being reduced, meaning that that treatment is killing those bacteria. And the same is true on the other graph, right? So both ways, it's just two different ways of showing almost the same thing, that when you combined the two, an antibiotic, and by the way, the concentration we used was what the microbiologists call MIC, which means minimal inhibitory concentration. So these are very low doses of antibiotic, below what would kill them in a test tube. When you combine those two things, the effect was lethal to the bacteria, lethal. So needless to say, that was good enough for me, almost. So my daughter didn't have MRSA. My daughter didn't have Pseudomonas aeruginosa. My daughter had Lyme disease, which is caused by Borrelia burgdorferi. So I got some Borrelia burgdorferi and started to grow it to the extent that it grew into a biofilm, which is not that hard to do in a laboratory setting because that's what they naturally do. And so what this shows, and I just really want to show you, over on the side there are pictures of the actual biofilm, right? So there are pictures of the biofilm, but here's what happened. The way we did this experiment very simply. We took the biofilms, had them grow on collagen-coated slides, and looked at them under the microscope for a long time. And so I had a student who was the most patient person on earth and thought this was fascinating and sat there and watched them for 3 hours. Seriously, looking at the microscope for 3 hours. So what he noted, and by the way, this went on in 2 separate rooms that were separated by 100 yards. All right, one where the pulse electric fields were being displayed, and the other one which was very, very far away from that, so that there would be no influence of the fields on the growth of the biofilm. And what we watched over time was basically the biofilm staying the same, or just looking the same at the end of those 3-hour period. But you'll notice that for the bacteria that were exposed to the electric fields, and this was at 150 kilohertz, the edges, the biofilm itself started to break apart. You notice that there were lots of these bubbles forming, which actually were not bubbles. They were the bacteria, because we have close-ups of these, which are not on this graph, showing that the bacteria were expanding in size and then actually exploding. So that's when I decided this is good enough for me, after I realized that technologically, this is very similar to what an MRI machine does. So you get into an MRI machine, you are exposed to magnetic waves, right, magnetic resonance, which actually just causes your bones and tissues and things to glow, which can then be imaged. So the energy is being let off as this energy that can be captured and imaged. And how many of you have spent time in an MRI machine? Is there anybody that hasn't spent time in an MRI? So the idea is this is safe, right? This type of treatment is actually safe if you also want to think that it's a frequency-specific type of effect. So if you have the right frequency, you're able to affect the bacteria. Our DNA is different, and so technically it should not affect our DNA as well. And given the amount of time that I spent in front of this machine, in front of that glowing tube while we were doing this experiment, I would have grown a second head by now if it was not. So all I wanted to do was to show you what I went through, the process I went through to decide that this was something that was useful and helpful. That there's many names for this technology, Rife technology. It also, the AMP coil technology goes this way. Anamed is another one that uses sound waves. I suspect there are others because the device my daughter uses called the GB4000, that is another version of it. So they are out there, they are available, and it doesn't require a prescription in order to have access to it. So thank you, and now the next person will come up and talk about their experience.